Welcome to our video about the fascinating world of Pokemon and their placement on the Tree of Life. Today, we are taking a closer look at Charizard and how it is related to Venusaur. Ready? Let's go! The last common ancestor of Charizard and Venusaur were the so-called amniotes. Amniotes are a large group of tetrapod representatives, characterized by their ability to reproduce completely independently of water, unlike amphibians. Instead of a short embryonic phase followed by a free-living fully aquatic larval stage, their early development involves a long embryonic or fetal phase without a free-living larval stage. The embryos or fetuses of the amniota develop in an amniotic sac filled with amniotic fluid, which is directly and completely surrounded by the amnion and more or less completely by other extraembryonic membranes. The amniota are considered a monophyletic taxon, meaning all extant and extinct species of this group derive from a single common ancestral species. The extant subgroups of amniotes include lizards and snakes, turtles, crocodiles, birds, and mammalia, mammals. The amniotic egg is considered a novel development and a key feature of the common ancestral species of all extant and extinct amniotes. It exhibits both modifications in the construction of the undeveloped, freshly laid egg and modifications in the development of the embryo compared to the egg and embryonic development of non-amniotic tetrapods. The ability to reproduce outside of water had the potential for early anamniotic terrestrial vertebrates to explore new habitats and thus avoid competition from other anamniotic species. With the development of the amniotic egg, as seen today in reptiles, birds, and monotreme mammals, this potential was realized. Terrestrial vertebrates were now able to spread into very dry regions and colonize the continents extensively. In placental mammals, this strategy has been significantly further developed. In these animals, the uterus is no longer the organ in which the eggshell is formed, but the organ in which the embryo and fetus develop within the mother's body until full term. During this process, part of the chorion fuses with part of the uterine wall to form the placenta, through which the embryo and fetus are nourished and excreted. In marsupials, the fetus does not mature in the uterus, but in a pouch equipped with a mammary gland. Due to these modifications in the reproductive mode, the eggs of true mammals are no longer macrolecithal/telelecithal, but yolk-poor with symmetrical distribution of formative and nutritional yolk. The first amniotes probably did not lay eggs with calcified shells. Such eggs are very poorly preserved as fossils. The oldest relatively clear evidence of an amniotic egg with an enclosed fetus comes from Mesosaurus, from the lower Permian of Uruguay. However, Mesosaurus fossils are found in large numbers in these aquatic sedimentary rocks, largely an anatomical connection. This means that the paleo environments there offered good conditions for fossil preservation, so the statistical likelihood of some Mesosaurus fetuses being preserved there is relatively high. The origin of the first amniotes probably took place in terrestrial, dry habitats, where conditions for fossil preservation were generally far less favorable. Furthermore, Mesosaurus is undisputedly considered a secondary, semi-aquatic reptile, which means that the amniotes must have undergone some adaptive radiation by the lower Permian. Indeed, in other regions of the world, North America, Europe, another unequivocal evolutionary line of amniotes is detectable at the end of the Carboniferous and in the lower Permian with, among others, herbivorous representatives that were already ecologically specialized. Therefore, determining when the first amniotes lived based solely on fossil evidence of amniotic eggs is challenging. A systematic examination and evaluation of the morphology of terrestrial vertebrate fossils provide clarity in this matter. The amniota are considered a crown group in modern, cladistically influenced systematics, defined by the most recent common ancestor, the hypothetical stem species, of all extant species that reproduce using the amniotic egg, or corresponding developments. By examining the morphology of recent amniotes and various fossil terrestrial vertebrate species, phylogenetic trees can be constructed, indicating which fossil species trace back to this common ancestor, even without fossilized amniotic eggs. The geologically oldest known species to meet this criterion are Hylonomus lielli, Protoclepsidrops hapless, Paleothyrus acadiana, and Archaeothyrus florensis, for rather small, lizard-like animals from the upper Carboniferous of Nova Scotia, Canada. Accordingly, the amniotes had already split into their two main lines before the appearance of these four species, the synapsids from which mammals evolved, and the sauropsids, from which today's reptiles and birds evolved. Indeed, according to analyses, Archaeopteryx is a representative of the synapsid line, and Hylonomus and Paleopteryx are representatives of the sauropsid line. Which of the two lines Protoclepsodrops belongs to is still unclear. The sauropsid line split into Eureptilia and Parareptilia must have already occurred, as Hylonomus and Paleopteryx are representatives of Eureptilia. The oldest known parareptile is Herpetonyx arsenalterum from the youngest Carboniferous. Now that we have learned a lot about the last common ancestor, let's delve into a more inclusive clade to which Charizard belongs, the Archosaurs. The Archosaur are a group of diapsid amniotes, which include crocodiles and birds as well as various fossil groups, including all non-avian dinosaurs and pterosauria. Archosaurs were the dominant vertebrates on land and in the air for much of the Mesozoic era. Today, they still constitute a significant part of the vertebrate fauna with over 10,000 species. The most important common features, synapomorphies, of archosaurs are found in the structure of the skull. Initially, the skull has 3 skull openings, and is therefore called a triapsid skull. The antorbital fenestra is located in front of the eye socket, while 2 more skull openings, the so-called temporal fenestrae, are located behind the eye socket. The 2 temporal fenestrae indicate that the archosaurs belong to the diapsids. However, this does not apply to living archosaurs today. They have highly specialized skulls that are no longer triapsid. Crocodiles have well-developed temporal fenestrae, but the antorbital fenestra is closed. In birds, the antorbital fenestra is only indistinctly separated from the eye socket, and the temporal fenestrae are small and fused with the eye socket. The lower jaw also originally has an opening, called the mandibular fenestra, which is still present in crocodiles. The teeth are thecodont, meaning they sit in individual sockets where they are connected to the bone by connective tissue, and they do not have multi-part roots. In modern birds and some non-avian dinosaur groups, the teeth have been reduced. Many purely fossil archosaur groups, Aetosaur, Phytosaurs, Ankylosaurs, as well as fossil and currently living crocodiles, have a back armored with bony plates embedded in the skin. The first occurrence of archosaurs is dated to the Upper Triassic for the crown group and the Upper Permian for the pan group. One of the oldest representatives of the Panarchosaur is Archosaurus rossicus from the Upper Permian. In the Lower Triassic, basal archosauriforms took over the role of synapsid apex predators from the groups Gorgonopsia and Titanosuchia that went extinct at the Permian-Triassic boundary. Another radiation occurred in the Upper Triassic with the emergence of dinosaurs and pterosaurs. In the Jurassic and Cretaceous, dinosaurs, in particular, brought forth an enormous diversity. During this period, archosaurs were the dominant group of terrestrial vertebrates. After the mass extinction at the Cretaceous-Paleogene boundary about 65 million years ago, at least one group of the dinosaur lineage, the birds, has survived to this day. The crocodile lineage took a completely different evolutionary path. Their representatives were increasingly displaced from their traditional terrestrial habitats by dinosaurs during the Upper Triassic and retreated to the water from the Jurassic onwards, developing the primitive, lizard-like appearance typical of modern crocodiles. Unlike our world, another group evolved from the archosaurs, the dragons. This is a highly diverse group that has conquered the oceans and the skies. Whether as sea serpents, wyrms, wyverns, or true dragons, they inhabit all habitats as apex predators. True dragons even developed 6 limbs, so besides 4 legs, they have an extra pair of wings. It is absolutely fascinating how the archosaur group managed to develop wings in 3 different ways. This is convergence and not a shared ancestor. Like the Aetosaur, Phytosaurs, and modern crocodiles, dragons also have thick, scaly skin that protects them from attackers. Now let's talk about Charizard's adaptations to the ecosystem in Kanto. With its massive wings, it can cover vast distances with relatively little energy expenditure. Charizards hunt alone and target relatively large prey like toros or rhydon. To avoid injuries while hunting, Charizard possesses an organ that produces fire, allowing it to breathe a massive fire attack at the right moment to immobilize its prey. That's it for Charizard, folks. I hope you enjoyed today's episode. In the next episode, we'll discuss Blastoise and why it's a strange third cousin of Charizard.